Today we will be focusing on insulin and glucagon, two peptide hormones produced by the endocrine pancreas that work together to regulate blood glucose homeostasis. The physiological mechanisms discussed are based entirely on the peer-reviewed article by Roeder, which integrates cellular, molecular, and systemic aspects of pancreatic hormone regulation. The objectives for today will be to identify the pancreatic cells responsible for insulin and glucagon secretion, explain how these hormones are regulated and maintain homeostasis, review cellular structures, explain signaling pathways, and we are going to discuss metabolic disorders such as diabetes mellitus. The endocrine pancreas is composed of clusters of hormone-secreting cells known as islets of Langerhans. In these islets, beta cells secrete insulin and alpha cells secrete glucagon. Together, these hormones antagonistically maintain blood glucose within a narrow physiological range. Roeder et al. Emphasizes glucose hormones depend on multiple factors: the hormone secretion, communication between different isolate cell types, and coordinated responses in order to change with metabolic conditions. Here we are looking at a macroscopic and microscopic view of the pancreas. At the gross anatomical level, the pancreas is located within the upper abdominal cavity and lies close to the duodenum of the small intestine shown here. The duodenum is the first segment of the small intestine and receives digestion secretions from the pancreas. Over here, you are able to see the proximity of the pancreas, shown here in yellow, to the spleen, which is located against the posterior abdomen wall. The pancreatic duct, shown here in green, transports digestive enzymes produced by the pancreas towards the intestine and joins with the duct to form the common bile duct that empties into the duodenum. The ductal system is important in highlighting the pancreas's exocrine role in digestion, delivering enzymes capable of breaking down carbohydrates, proteins, and lipids. Inside of the pancreatic tissue are acinar cells, shown here in yellow. Acinar cells form exocrine acini and secrete digestive enzymes including amylases, proteases, and lipases into the pancreatic ductal system. Although acinar cells are not all hormone-secreting cells, the location of them and their function are important for understanding how the pancreas integrates digestion with metabolic regulation. Over here, we have pancreatic isolate cells. And following this arrow, we now have a zoomed in version of the pancreatic isolate cell. This shows the exocrine acini surrounding the isolate with the physical relation between the endocrine and the exocrine tissues. The beta cells, shown here, secrete insulin and are able to be seen very prominently. They are the large white dots. Insulin lowers the blood sugar by promoting glucose uptake, glycogen synthesis, and lipid storage in peripheral tissues. Beta cells also create C-peptide, which is released in equal amounts with insulin to serve as a marker for the endogenous insulin production. Amylin is also a hormone to slow down gastric emptying and suppress the postpartum glucagon secretion, which is already released via the beta cells. Alpha cells are known to be the source of glucagon, which can raise the blood pressure by stimulating glycogenolysis and gluconeogenesis inside of the liver. The position of the alpha cells within the pancreatic isolate also allows a fast response when an individual is experiencing hypoglycemic conditions due to the increase in the blood glucose levels. Somatostatin are created by the delta cells, which, although not shown here, is a pancreas hormone to inhibit insulin and glucagon secretion. Somatostatin acts locally inside of the isolate to tune hormonal output, preventing excessive fluctuations within the blood glucose level. F cells or PP cells, which would be the pancreatic polypeptide, regulate the pancreatic exocrine secretion to influence the gastrointestinal motility and appetite regulation. Epsilon cells create the hormone ghrelin, which is involved in the appetite stimulation and energy balance, in turn giving the coordination and signals related to hunger and or energy availability. The arrangement shown here allows all pancreatic hormones, insulin, glucagon, amylin, somatostatin, pancreatic polypeptide, and ghrelin to be quickly distributed throughout the body to maintain glucose homeostasis. Here we have figure 2. Figure 2 is going to demonstrate the dynamic homeostasis regulation of blood glucose levels throughout the antagonistic actions of insulin and glucagon, centered around the average glucose levels of 4 to 6 millimolars per liter. We're going to start here on the left side of our screen with our burger, our fries, and our soda. This food is going to represent the food intake into the system as a postprandial state. When the food is intake to the system, the blood glucose levels are going to rise, shown here as the blood glucose with the arrow increasing. This increase is to show the glucose is detected in the pancreatic beta cells, bringing us to the pancreas. Because now the pancreas knows to secrete that insulin due to the elevated blood glucose, shown here as insulin. It is then able to act on multiple targets, as we see here, because we have two different arrows in skeletal tissue and adipose tissue. The insulin is going to increase the glucose uptake, shown here by glucose uptake, increase in arrow, by insulin stimulating translocation of glucose transporters to the cell membrane. On the other hand, the other target is going to be the liver, where insulin is going to promote glycogenesis, so there is a conversion occurring here. The glucose is then turned into glycogen for storage while inhibiting glucose neogenesis. As a result of the coordination on the muscle, adipose tissue, and liver, blood glucose levels then are able to drop back and return to where they were once as a normal level. The balance scale seen here in the circle is a visual representation of reinforcing the restoration of glucose homeostasis. We then have the fasting state, known as the post-absorptive state. This is signaled here with the nighttime sky. Blood glucose levels are going to fall due to the detection of the pancreatic alpha cells, represented as the pancreas here. When the pancreas secretes glucagon, and the glucagon targets mainly the liver, which is then going to stimulate glycogenolysis, which is the breakdown of stored glycogen into glucose. And it's also going to do glyconeogenesis, which is the synthesis of new glucose from non-carbohydrate substrates. The glucose produced within the liver is then going to be released back into the bloodstream, causing the blood glucose levels to rise back to a normal level. Therefore, once normal glucose levels are restored, glucagon secretion is then reduced through the negative feedback. This circular pathway shows that this is a continuous and a dynamic feedback system versus just a one-time response, showing insulin and glucagon act as antagonistic hormones. Our next discussion is going to be discussing the molecular and the electrophysiological mechanisms of glucose-stimulated insulin secretion, GSIS, in pancreatic beta cells with glucose metabolism, ion channel activity, calcium signaling, and vesicle exocytosis. This process begins at the beta cell plasma membrane, where glucose is then entered in the cell through the GLUT2 transporter, labeled here in blue. The GLUT2 is a high-capacity glucose transporter, which allows glucose to influx increase proportionally with rising blood glucose concentrations. When glucose is inside of the cell, metabolic processing begins to show here as the metabolism, which then leads to mitochondrial oxidation. The metabolic activity then increases the ATP and ADP ratio, which is important for the signal linking glucose availability to the electrical activity. With an increase in ATP and ADP ratio, this is affecting the ATP-sensitive potassium, also known as the KATP channel, which is made of Cr6.2 pore-forming subunits and the SIR1 regulatory subunit. With the ATP increasing, the KATP channels will close, reducing the potassium ion efflux from the beta cell, shown here as this red arrow. And this is going to be a decrease in the potassium ion movement, shown here with the X. When the KATP channels close, the membrane then depolarizes, which changes the electrical state of the cell membrane, triggering an opening of the voltage-dependent calcium channels known as the VDCCs, which is shown here in purple. When the VDCCs open, calcium ions are then able to enter the cell, allowing an intracellular calcium concentration. This increase, shown here as a calcium ion plus, is the immediate trigger for insulin secretion. Over here, we have a zoomed in version. So the LDCVs in insulin large dense core vesicles move toward the plasma membrane and cluster near the membrane in a response to elevated calcium ion levels. Here is the details of that influx. So the snare protein machinery is responsible for fusion and exocytosis with the help of VAMP on the vesicle membrane, SNAP25 on the plasma membrane. The snare complex, which then pulls the vesicle and the plasma membrane together. Synaptogomin is a calcium sensing protein located on the vesicle membrane, which triggers to a rapid membrane fusion, as seen here, when calcium rises. When fusion occurs, the insulin is able to then be released into the extracellular space down below, which is going to be this space here, shown as insulin release, which then allows the insulin to enter the bloodstream to the peripheral tissues, lowering the blood glucose level. This figure is a perfect example of how glucose metabolism is electrically and chemically coupled to insulin, exocytosis, nutrient sensing, ion channel regulation, calcium signaling, and vesicle fusion into a controlled endocrine response. This figure from Roeder shows the integrated multi-organ regulation of insulin and glucagon secretion, and the actions focusing on glucose homeostasis being controlled through coordination between the pancreas, the brain, the liver, gut, adipose tissue, and skeletal muscles. The green arrows throughout this figure here will represent the stimulatory effects, while the red arrows shown throughout this figure will represent the inhibitory effects. We're going to start here with our adipose and our skeletal tissue. Starting with the adipose tissue, leptin, secreted by the adipose tissue, is going to exert an inhibitory effect on the insulin secretion by signaling long-term energy to the pancreas and the central nervous system, also known as CNS, suppressing excessive insulin release. Adiponectin, which is also released from adipose tissue, has a stimulatory effect on the insulin sensitivity, supporting the beta cell function. Interleukin 6, IL-6, is then released from the skeletal muscle during contraction, which stimulating pancreatic endocrine responses, linking physical activity to glucose regulation. Next, we have the pancreas secreting the insulin and the glucagon. Insulin is produced by the beta cells, which is the primary glucose-lowering hormone, and glucagon is produced via alpha cells acting as a glucose-raising hormone. This arrow here is going to show the beta cell growth, which is the insulin's autocrine and paracrine roles in supporting the beta cell survival in the correct metabolic conditions. Down here, we have our gut isolate axis with nutrients entering in. This is going to show the role of the gastrointestinal tract in the postprandial glucose regulation, with nutrients entering the gut to stimulate enteroendocrine cells like K cells, L cells, G cells, and I cells. All of these cells secrete hormones like GLP-1 and GIP, which are going to exert a stimulatory effect on the insulin secretion and an inhibitory effect on the glucose secretion. CCK and gastrin are pancreatic hormone secretion, giving that linkage to the digestive and endocrine responses. Our brain isolate axis shows the CNS control over the pancreas hormone secretion. The hypothalamus includes nutrient, hormonal, and neural signals, while the hypothalamic neuropeptides such as NYP, melacorpus, POMC, MCH, VIP, PACAP, and CGRP all exert stimulatory and inhibitory effects on the insulin secretion, shown here in the red and the green arrows. The neurotropic factor BDNF enhances the stimulatory factor and beta cell function. Finally, the cephalic response anticipates insulin release triggers by sensory cues like the smell and the sight of food prior to the nutrient absorption. So we need this response here before we can get the nutrients here. And our liver isolate axis is going to show the insulin's metabolic actions within the hepatic pathways. Our insulin stimulates, which are our green arrows, glycolysis and glycogenesis, while promoting glucose utilization and storage within the liver. However, on the other hand, glycogenolysis, shown here, and gluconeogenesis are going to prevent hepatic glucose output. Bringing all of what we just learned into our clinical relevance, it's important to know disruption of insulin and glucagon can lead to disorders within the human body relating to glucose homeostasis, specifically diabetes mellitus, hypoglycemia, hyperglycemia, and a hormonal imbalance. Type 1 diabetes is an autoimmune of pancreatic beta cells, resulting in an absolute deficiency of insulin. Without insulin, the uptake of glucose by the skeletal muscle and the adipose tissue will be decreased, resulting in the hepatic glucose production being increased. This is due to the unopposed glucagon action, leading to constant hyperglycemia and an increased reliance on other pathways like lipid metabolism, which can lead to ketoacidosis if left untreated. However, in type 2 diabetes, insulin is still produced, but peripheral tissues become insulin resistant, which allows the beta function to decline over time. The XX glucagon secretion plays a part in the fasting and the hyperglycemia. Although blood glucose may be elevated, glucagon will still stimulate the hepatic gluconeogenesis and glucogenolysis, showing diabetes is not just insulin-related disease, but can also be based on a hormone dysregulation. Hypoglycemia is when there is an imbalance in the blood glucose and it falls below the normal physiological ranges. Under average conditions, decreased glucose levels stimulate glucagon secretion to restore blood glucose through the hepatic glucose production. But when this response is damaged or impaired, the risk of severe hypoglycemia increases with long-standing diabetes. These conditions show the importance between insulin and the glucagon versus just a singular hormone. Disruption among the system, no matter how small, shows insulin deficiency, insulin resistance, or wrong glucagon signaling can lead to a significant metabolic disease.